Now, as I said in the first glycolysis lecture, we usually only talk about glucose because the model for this, because glucose starts at step one. What we found is that pretty much everything you ingest, food-wise, can somehow fit into the whole cellular respiration thing. You look up above here. The carbohydrates, some of the amino acids, and glycerol can fit into glycolysis. Like I said, the carbohydrates, glucose, fructose. Fructose, remember that was like the third step of glycolysis. The carbohydrates, the long chain polysaccharides get broken down glucose by glucose. Some of the amino acids can fit in here because remember they have their R groups can be a chain of carbons. Glycerol, which is part of the fatty acid molecule, is a 3-carbon. So with some modifications, it can fit into the second half of glycolysis. If you go back and you look at, let's say the citric acid cycle, you'll look at some of those steps there, churning away citrate, isocitrate, amino acids. The other steps involved there look quite similar to amino acids, so many of our amino acids can go right in there. The fatty acids, long chains of carbons, get cut every 3, fed into pyruvate oxidation, cut every 2, fed into the citric acid cycle. Side note, the whole idea of some of the fad diets we've seen over the past decade or so, the Atkins, the keto diet, the low carb diet, is trying to force a burden on cells. At the cellular level, it's trying to force a burden on the cellular metabolism by getting rid of glycolysis and going straight to citric acid cycle. Maybe pyruvate oxidation. You lower what's happening. It takes more energy to prep the amino acids, cut them down, so there's that. So when you look at the citric acid cycle and you see how things feed in, how things can feed out. Remember, this is a churning mass, not a pathway. So you see arrows pointing in, arrows pointing out. Acetyl-CoA can be turned into acetoacetate, which can then be the building block for leucine, lysine, phenylalanine, tyrosine. Oxaloacetate can be used as the foundation for the making of asparagine or aspartate. Succinyl-CoA, with some modifications, can become isoleucine, valine, methionine, threonine. Those amino acids, with some modifications, can become succinyl-CoA and go right into the citric acid cycle. Now, as in all things biological, we don't just start the ball rolling down the hill and hope for the best. We like to have a series of steps that are going to be under control, some sort of regulation. Cellular respiration is also going to be under some control. We find that the control of cellular respiration can be at the level of hormones. Hormone level can increase, decrease glucose availability. Increase, decrease glucose availability for whether or not glycolysis can occur. Enzyme reversibility. Sometimes we saw arrows going one way, arrows going back. Sometimes enzymes are going to be switched on, switched off. Substrate now becomes a product. Product is going to be switched back to the substrate. Lactic acid buildup is going to shut down some things. Feedback controls, as we talked about before. Feedback inhibition. When you look at insulin, insulin is a good one. Insulin floating by in the bloodstream and the interstitial fluid will bind to the insulin receptor on your cells and will cause for the uptake, the increased uptake of glucose. So if your insulin levels are high, means your blood glucose levels are going to suddenly start to drop. Why are they dropping? Because all the cells are taking up the glucose. It's immediately going to glycolysis. It's immediately going to be fed into cellular respiration. And when you look at the different pathways, glycolysis, citric acid cycle, the electron transport chain, the different phases, pathways, tomato, tomato here, are going to have enzymes in them that will be affected, turned on, turned off. Their activity increased, decreased based on some substrate. Say citric acid cycle, CAC. The isocitrate dehydrogenase, one of the earlier steps. Its activity will be increased due to a low level or a high level of ADP, A diphosphate. If A diphosphate levels are high, that means A triphosphate, ATP levels, are low. So A diphosphate is going to increase the activity of this enzyme. If A triphosphate levels are high, well, it's going to turn down, slow down that mixture, that mishmash of enzymes that known as the citric acid cycle. Pump the brakes a bit, slow it down, so that way it's not going to burn through all the ADP. You're not going to have all this ATP that the cell doesn't really need at this moment.